I was getting ready to come to this conference, and I had told Alyssa I would do her talk for her, and then the schedule started coming out, I said, oh my gosh, I have my own talk. I didn't realize. I think I had submitted an abstract to one of the previous renditions of the conference, and it got carried over, and I hadn't realized it. So you get to have me here for another 15 minutes. I hope that's all right with you. Okay, so this is more focused on the work that I've been doing for many years. I started this as a postdoc at the Smithsonian Marine Station 11 years ago. I can't believe I've been there that long. But I've worked on coral settlement queues for many years. I've recently started collaborating with Margaret at Sea Corps and some other people to try to see if we can use this to apply to restoration purposes. So that's what I'm going to talk about now. Okay, so here, tetrabromoperol, TBP. You guys will get very familiar with this TBP. I'm going to say it a lot. Tetrabromoperol is a compound that induces settlement. We published on this back in 2014 in this Proceedings of the Royal Society paper, and I can provide that to anybody who's interested. This compound is produced by a bacterium. This is produced by a Pseudoalteromonas bacterium, and as Nikki was saying, she's finding some of those, some bacteria in that genera in her potential probiotics that she's looking at. This is a genus that's really known to produce a lot of interesting compounds and have a lot of activity in the marine environment. So these bacterium, or this particular bacterium strain, was isolated from a crustose coralline algae called Paragonia lithonsolubility. And so this is just kind of, I'm going to quickly go through a summary of what was in that 2014 paper as the background, but basically we looked at whether this compound would induce settlement in 3 different species of corals. So the first one here is Parietes asteroides, and what you're looking at on the y-axis is the amount of settlement, and so that's completely attached and metamorphosed. And then across the x-axis are different concentrations of tetrabromoperol, so it goes from 0 on the left all the way up to 500 nanograms per milliliter. And then the very last bar on these graphs is going to be in comparison to the CCA Hydrolithon borgesonii, which is a pretty well-known CCA that induces settlement in a lot of Caribbean corals. So what pops out here is that we have, settlement on CCA at around 46 to 60% here. And when we add just tetrabromoperol, these are in sterile seawater in a 6-well plate, we get as much settlement or more as we do with the CCA itself. Same thing with Acropora palmata and Orbicella franksi. Here you can see the response to the CCA versus the response to the tetrabromoperol at its sort of optimum concentration. Okay, so the pandemic's been rough on all of us, but on my research, it's been kind of good. Because we couldn't travel to do spawning ourselves, we've created some really great relationships with people throughout Florida who have been really generous in providing us larvae. And that's allowed us to test a lot more coral species than we would have been able to otherwise. And so at this point, I've tested tetrabromoperol with 12 different Caribbean coral species, and it induces settlement in all of them. This figure across the bottom, it's basically showing you a number of those different coral species. The bar on the very left of each one of those graphs is a control. It's just a solvent control, DMSO. Then filter sterilized seawater if you don't give it anything. And then 2 different concentrations of TBP there that's showing. But basically, you can see that TBP is inducing settlement in all of these things. Including, we recently tested it with Astrangia poculata, which is a temperate species. It also induces settlement in Astrangia poculata. So we're looking now at 9 different genera, 7 different families, 2 different life history stages. It induces settlement in everything I've given it to. We also then have been looking at related compounds. So one of the downsides of tetrabromoperol, it's kind of a double-edged sword, but it's not stable. So it does break down fairly quickly over time, which could be good for application in the environment because it's gone very quickly. But at the same time, it can be challenging for trying to use it in some of these situations. So we've been looking for other options that may be more stable. We've tested a number of different compounds. These are compounds that are part of the biosynthetic pathway of tetrabromoperol. And we tested these with Acropora palmata and Porites asteroides in 2016, and none of them induced settlement. Tetrabromoprol is the only one of these 6 compounds that induces settlement. And you can see even this one down here, tribromoprol, is only different in 1 bromine. It's missing 1 bromine, and it doesn't do anything. So this past couple years, we've tested another suite of compounds with a whole larger group of different corals. Same thing. So that first compound there is tetrabromoprol. So those first 2 columns are 2 concentrations of tetrabromoprol. And you can see, fairly high numbers in both of those columns all the way down through all those different coral species. And then if you look anywhere past that on that figure, it's nothing more than 4%, which is not significant. And you can see how similar those compounds are, right? They all look very similar. You change a bromine to a chlorine, nothing. You add a methyl group, nothing. It's very, very specific. So I think it's very interesting. And as I said, we're trying to figure this out now if this is applicable to restoration purposes. So we've been working with Margaret and the people at Secor, seeing if we can dose her tiles in her cribs with TBP to induce settlement on those tiles and sort of get away from the model of taking tiles offshore or putting them somewhere to condition for several weeks, getting variable conditioning with organisms you're not really in control of, and that sort of thing. So we're trying to see if we can use this compound to induce settlement at these situations without having to go through that conditioning process. And we have a little bit of promising results in that way. So these are some data from last summer, just very preliminary data that Margaret collected. These were some bucket assays, so these were done in her crib, but in buckets instead of just the entire crib. She had tiles that had been conditioned for, I believe, 4 weeks, something like that, and she compared those to tiles that had not been conditioned at all, but had TBP added to them. And so you can see in that first figure that the number of settlers on the TBP tiles are lower. But if you saw some of the talks this morning, we heard that maybe having more settlers per tile is not always better because there's some mortality that happens after that if you have too many on one tile. So we think that's a kind of an acceptable range of number of settlers per tile. But the really interesting thing on the right side is that these are the number or the proportion of tiles that had 0 settlers on them. So really for restoration, you want to have tiles. You want every tile to have a baby on it, right? So these tiles, you can just see there's a number of tiles in the positive controls, those that were biofilmed, that didn't have any settlers. Whereas in the TBP treatments, there were very few that had no settlers. Almost every tile had a settler. So that's very promising. So this was done with Orbicella fabulata. We're hoping to scale this up to the crib level. We've been working on this a little bit, but of course, it's coral larvae and things are challenging. It's always tricky, but we're working on that and working on different ideas for ways to utilize this really strong signal for restoration purposes. So some of the areas that we're looking toward in the future are, again, enhancing the ability of TBP for coral restoration, looking at ways to stabilize the compound, incorporating it into settlement substrates in some way that it could maybe, be released like a slow release pill, something like that, those sort of things. And then just from a science perspective, I'm really interested in looking at whether this is really a natural settlement cue. Is it there in nature? Is it what corals are really cueing in on the reef? Like, are we seeing it produced in amounts that they would respond to? Do we see that really happening? Because this is mostly lab-based, it's really completely lab-based. And so that's really interesting. And then I'm really interested in trying to understand the mechanisms behind it. So how and why is this compound so active? So I'm really open to collaborations on these questions. If anybody wants to get in touch with me, I'd love to work on them. I'm very interested in those questions. And with that, I want to thank the whole team that's been working on this work. Again, like I said, Lily is here. I have a couple interns, a couple techs that have been working on this. My postdoc, Dr. Alyssa Dimpko, of course, Val Paul, and I really need to thank the people who have been providing us larvae, which is amazing. So we have Carrie O'Neill at Florida Aquarium, Joanna from NOVA, Dana Williams from NOAA, Amanda Bork from the Biscayne National Park. They all provided larvae for this project. And then our collaborator at Georgia Tech who produces the TBP for us, Vinny Agarwal. And our funding provided by Secor, Florida DEP, and OceanKind. And with that, I will answer any questions. I think I made up time. That's what happens when you're giving your own talk versus somebody else's talk. Yeah, you can ask on either one. I'm happy to answer. Yeah. So the question was, as a resource practitioner, restoration practitioner, how do you get your own TBP? Well, I would say, what would I say to that? I would say, this is obviously still in development, and I'm not sure I would recommend it just be used yet. We're still evaluating it. I'm not sure I would recommend it just be going out to people to use indiscriminately at this point. But I think that it's probably I'm not a synthesis chemist, right? But I would say it's probably easy to synthesize. So if you found a synthetic chemist, they could probably make it for you. Don't buy it from Sigma Aldrich because it's not there. I bought it from Sigma Aldrich and it was completely decomposed by the time I had it. There was no TBP left in what they sent me. So just FYI. I think that slowed a lot. I know it slowed people down. It slowed me down. It slowed another colleague down. They get the compound from Sigma Aldrich. They think it is what it's supposed to be, and then it's not doing anything. And then you're like, well, what's going on? But so I'll put out that caveat: don't buy it from Sigma Aldrich. Yeah.
How are you applying it to your settlement tiles, and then do you know how long it lasts on your settlement tiles?
Those are great questions. So she asked, how are we applying it to the settlement tiles, and how long does it last? So we're exploring methods for applying it to the tiles with very limited success at this point. Our best success has actually been just adding it directly to the water. So in those bucket assays that we did with Margaret, we just pipetted it right into the water, and those were the results that we got. So that was really promising because we've been working for years being like, how can we get this on the tiles? And I was like, oh no, we just squirt it in the water, it's fine. So fine, Margaret's shaking her head like, kind of, yeah, we need a lot more work on that because we're not sure, but that was, I was very impressed with those results. Yeah. It's you, yeah, Sheila.
Question from Glasgow.
Uh-huh.
I'm always really curious to know if after the survivorship assays, did you sequence the recruits or anything to see if that whatever put in them is different and how the results are different?
So the question was after we treated the corals with probiotics and ran the assays with them, did we do anything to sequence the corals at the end? The answer is no. We have taken samples, though, with the idea that if funding, time, whatever is available, that we could do some of that sort of thing. Joanna?
Things that are usually good settlement use, it means that they're going to be useful for the coral later on. So any ideas in terms of how it will affect survival and growth, and how do you think that this compound what is it that this compound promotes? Immunity? Is it
Mm-hmm.
what is happening?
Yeah, so the question is sort of, do I think that tetrabromoprol is providing some sort of advantage to the corals, and what might that be? I'm not sure if you were here for the talk that I gave just before this, but we're starting to think they might provide some protection against disease, which I didn't really expect, but that was just a side effect of some work that we've been doing, which again is very preliminary, and we need to really look into that some more. I suspect that, so we know this compound has antibiotic properties, so I suspect that it may be altering the microbial community on the substrate in some way that's beneficial to the corals. That's my guess, but again, this is not really been touched at all, so that's a lot of area that I want to move into, is trying to figure out what's happening in the environment. Is this compound really being released in ways that corals can detect it? Evidently, we need to talk about this later. Thank you all so much for listening to me for a full half hour. And I will release the stage now.